Okay, so our last video was on proteins and their structure and some of the functions they undertake within a cell. So we're just going to look at further details of protein structure, and we're going to start looking at the levels of protein structure. So protein structure comes on different levels, building from a 2D sort of structure to a almost 4D sort of interlocking structure between different subunits and stuff. So that's what we're going to look at. So we'll dive right in, and our first the first type of structure we're going to look at is the primary structure. So the primary structure is pretty straightforward. It's just the amino acid sequence of a peptide chain. So if you have alanine, I think here we have valine, glycine, and then tyrosine in a tripeptide. But if you keep going, if we had valine, glycine, tyrosine, generally for introductory biology, you don't have to memorize the amino acids. You will be given a table if you need to know them. But it could keep going: asparagine, isoleucine, leucine, aspartate, on and on and on. You can have thousands of amino acids. The primary structure is their specific sequence, and most importantly, the sequence of their side chains. So obviously, the side chains are the variable regions between the amino acids, and while the main chain has a lot to do with secondary and tertiary structure, as we'll look at next, it's the side chains that are involved in most of the binding and catalytic activity of proteins, and also involved heavily in determining tertiary and quaternary structure, which we'll look at now. So secondary structure is quite crucial when looking at proteins, and when you look at secondary structure, you look at a couple of main motifs within secondary structure. So secondary structure is when you start taking this chain and building it up into a three-dimensional object. And there's two main ways you can do this. Amino acids packed together generally either in this alpha helix, which results from hydrogen bonding between residues 3 amino acids apart, and they form a helix, or a screw-like pattern. So here's our first amino acid, and going around up, we're going up the screw sort of thing. The chain continues there. We have the next one, and then so that's the first one, then the second one, and the third one after our original amino acid is directly above our original amino acid, and you get hydrogen bonding. So what happens is when you have a peptide bond, you have this sort of arrangement here. This is very polar, so you have the double oxygen, so that's a bit negative, and then you have this nitrogen and hydrogen here, and that's slightly positive, and you can get hydrogen bonding between these two groups, sort of thing. So the double oxygen below will hydrogen bond up here, and you get hydrogen bonding between the carboxylic acid, between the carboxyl group on one amino acid and the amine group on an adjacent amino acid, and that forms these hydrogen bonds in here between these adjacent amino acids in this helix, and that gives this alpha helix strength and gives it its structure, this highly regular bonding between adjacent amino acids 3 residues apart. And I've tried to magnify a bit. The alpha helix actually has characteristic bond lengths between atoms, and hydrogen bond lengths, so like spacing between layers, and those you don't have to memorize them or anything, but they're important parameters researchers use when when investigating the structure of proteins. So along with the alpha helix, there's one other main type of secondary structure motif, and that's beta sheet. So in the beta sheet, instead of in a helical pattern, the strands are adjacent to each other, and they run in sort of a wave-like pattern. So we have amino acid here, an amino acid here, and this is the chain of amino acids going along, and the chains stack next to each other in a highly regular fashion, and you get hydrogen bonding between these adjacent chains, and you form quite a flat structure, as opposed to the alpha helix, which makes a sort of cylindrical cylinder. And each of these have different applications within a protein. Alpha helices are quite often found in DNA binding motifs, so two helices fit very well into the grooves of DNA, and they bind to the structure of DNA quite well. And beta sheets are found in a lot of structural proteins, where you have this very strong intermolecular packing between molecules. So moving on from secondary structure, we have tertiary structure. So tertiary structure is, just going to adjust the brightness. So tertiary structure is extremely important because we're building up further into a three-dimensional functional protein, and it's important that we discuss things called domains. So different proteins can have different segments of proteins, of the one protein, that contribute to different things. The main example of this is where you have a membrane protein, so it goes through a membrane. It'll have an extracellular component, then it'll have a hydrophobic region embedded in the membrane, and it'll have a intracellular component. So we have these. So we have these three components here. We have an extracellular component, an intermembrane component, and an intracellular component, and these are each separate domains. So the extracellular domain, and they each have different characteristics. So both the intracellular and extracellular domains will have a lot of hydrophilic amino acids which interact with the aqueous environment outside here. But in the transmembrane domain of the protein, you'll have a lot of hydrophobic residues, which sit easily and nicely in the hydrophobic region of the fatty acids in the membrane. So different domains are something that come along with tertiary structure. You also get tertiary structure maintained by disulfide and salt bridges. So disulfide bridges form between cysteine residues, which have their functional group ends with a HS group. So a sulfur and a hydrogen. And when you have two of these adjacent to each other, you can reduce down. So we have one chain here and one chain here, and you get this disulfide bond. So the sulfur groups bond to each other, and the hydrogens are kicked out in a dehydration reaction sort of thing. And these two sulfides bonded to each other form a tight covalent bond between two sections of the polypeptide chain about here and here. So this would be the polypeptide chain, this would be the polypeptide chain. There's other stuff in the cysteine residues, but I'm just keeping the sulfur to simplify it. And by forming this covalent link, they can strongly bind two areas together. Similar with salt bridges, I discussed charged amino acids in the last lecture. They can bond with an oppositely charged amino acid residue side chain, and that forms a salt bridge, which is also a very strong linkage between two parts of a chain. So tertiary structure has a lot to, because it's building up from the individual sort of 3D subunit motifs of secondary structure into a full 3D structure. Catalytic activity is highly dependent on it, and you also can see two main types of tertiary structure, and that's globular or fibrous. So globular is where you have a protein like myoglobin, which is, it looks roughly spherical, and its chains all arrange to be tightly packed into a sort of a glob. It's fairly onomatopoetical, I guess. Whereas fibrous proteins such as keratin or actin, which are actin's part of the cellular cytoskeleton, and keratin is part of your hair and nails, they arrange into highly crystalline sort of bundles of protein, and they're thread-like sort of thing, and they're like fibers, and those are two distinct main classifications for proteins. But then again, you have things like these transmembrane proteins, which have vastly different appearances, and so this isn't all inclusive, but it's mainly how you could class two types of proteins. Moving on to the last level of structure, so we go primary, secondary, tertiary, somewhat like education levels. Then we move on to quaternary structure. So this is where you combine multiple polypeptide chains into a single protein. So you have multiple subunits which are bonded covalently, usually through disulfide or salt bridges, so strongly bonded to each other, and they can display a bunch of different so like hemoglobin, a normal enzyme, its kinetics are fairly straightforward. Like it only has a certain effectiveness, and that's sort of set. Whereas with enzymes like hemoglobin, you get this trait called cooperativity, where hemoglobin, when it binds oxygen, it has 4 subunits. So as each of the subunits bind an oxygen, it actually makes the other subunits more capable to bind oxygen. So quaternary structure, different quaternary structure having multiple subunits in a single protein can have some very effective it can make some very effective changes to the function of the protein and help it in its role, such as hemoglobin as an oxygen carrier. We'll look at hemoglobin's cooperativity in further lectures on kinetics, but it's just an interesting example of how quaternary structure really does affect the function. And then past quaternary structure, you can have further modifications of a protein. So you can have phosphorylation, which is generally involved in activating a protein. Glycosylation is generally involved in a bunch of other things, recognition by another protein and such. You can have cleavage, so a polypeptide chain can be cleaved to get rid of certain segments which were markers for things before, say, but weren't needed after a certain point. And then you can have adding of prosthetic groups like heme groups in hemoglobin, and other groups that involve, say, inorganic groups which help the activity and function of the enzyme or protein. So that's the levels of structure: primary, secondary, tertiary, and quaternary. If you need to remember the flow, it does help to remember like education levels: primary school, secondary school, tertiary education is university, and then quaternary going on from that. That's a useful mind trick you can use, or but mainly you will need to be well rehearsed on things about secondary structure like alpha helices and beta sheets, because they're very important building blocks of proteins that you need to really understand.